Okay, so let's look into glycosidic bonds. So this is one, correct? 2, 3, 4, 5, and 6. I hope you guys see the carbons. Now I'll draw another of this. And what is this? Is this alpha or beta? Right. So this is an alpha. Which of these two molecules, they are coming in together like this, right? Which of the two OH will start to come together and remove their water? Which carbons, or OH at which carbons will come together and remove the water? Tell me the carbon numbers. And just reminding you, this is alpha, this is also alpha. Look at the structure. So there's a reason I'm bringing these two things closer. Which of these OHs do you think will come together and remove the water? If one molecule of glucose, an alpha D glucose is coming in and interacting with an alpha D glucose here, which two OHs will come together? This and this. In order to understand that, we'll have to look into the entire structure of the sugar, in which planes they are coming, how these planes are interacting and all that. But what in nature in general we see that this interaction is very common. Maybe there could be other glycosidic linkages as well. But this 1,4 linkage between these two alphas is very common. That is why I have taken this example, because this is very common. So what I can call this is a 1,4 glycosidic linkage. But now you have to understand why this is called 1,4, because the linkage is between a 1 and a 4. So if you draw the structure of the resulting compound, I'm just trying to, so this is how it is represented. Now in here you don't have carbons, okay? You don't have carbons. So I'm just representing this to show that the bonding is happening below the plane of that. Right? So like both these are coming from below the plane. And that's what I'm trying to show in here using this linkage. This, if you keep on, so this can happen to, so we brought 2, but you can bring multiple, right? This kind of linkage in which you have 2 alpha groups coming together and doing a glycosidic, so this bond between 2 OHs in 2 glucose molecules is called a glycosidic bond. This if you keep on continuing, so if you continue to do this, and this is how we represent it. So this represents the polymer is going on and on. So you can form all these groups on either sides, in which I'm only showing these two groups, these two monomers. The molecule that will form when you have n number of this, or like a very high number of this, is called a starch molecule. Probably you have heard this term, starch. Where do you have starch? Where have you heard about this term? Do you eat starch? You don't eat starch? So what starch do you eat? Amber, what starch? Which food will have starch? Potatoes, rice, wheat, all these things will have this starch. So all that what you are eating, so is this polymer between all these glucose. That's what it is. And glucose is that simplest molecule, and you form these, and that is the starch. That is the energy storing molecule, right? So this is an energy storing. You get a lot of energy by eating these. So how will you get energy? Now, when your body will process this, it will start breaking those bonds. It will start breaking these bonds to release that glucose, because then it will have to break down glucose, and then you get that energy out from the glucose, right? So you have to break these bonds. And our body can actually break this bond, there is no problem. Okay, so this is starch. But let's look at another molecule in which one of them is alpha, but the other is beta. Right? So one of them is an alpha glucose, the other becomes a beta glucose, and let's try to see what happens with that. So this is beta because this is cis to this group, right? Now this is going to come together Now, when you bring these two things together, now you can see that these two are coming from opposite planes. So one of them is alpha, one of them is beta. So now what will happen? You will form these slightly different molecule. So now this is what you start getting. You can see that this is being represented by this weird looking thing. These are not carbons, but this shows that they are coming from these different planes. So one of that OH was somewhere above and this was below. So this is different, right? So this thing is going to give rise, and if you make these kinds of linkages, you get cellulose. Just by a slight difference in the orientation of these glycosidic bonds can actually produce very different type of molecules. One is starch, the other is cellulose. Simplistically, you can understand, have you heard about this term cellulose? Bark of trees. So all that is cellulose. Humans cannot digest cellulose. We do not have the enzymes, or we don't have the capability to break down the bonds in cellulose to release those sugars. So we cannot digest them. So just imagine, just by a small change in the orientation of these bonds is giving rise to planetary scale or biosphere scale changes just by doing that. And cellulose is one of, I think it is the most abundant biomolecule on Earth. Cellulose. Very stable, very difficult. So like you cannot, human beings cannot digest cellulose, but termites can. So there are certain enzymes or symbionts, as we call it, so that live in termite gut that can actually break this down, and that's why they can eat all this wood. We can't. So that's why. It's the different kind of chemistry that they have in their digestive systems. Okay. Now, okay, that is the deal with the sugars. Now, with certain small differences, you can have, so this was the normal glycosidic bond. Now, the glycosidic bonds could also have slight variations in which not two OH and OH are coming together, but you can have N-glycosidic bonds and you can have S-glycosidic bonds. So, so you have sugars, you can have other molecules which might have an NH2 group in it or an SH group in it. So these sugars will have these OH groups. So you can either find an N-glycosidic bond or you can have S-glycosidic bond. But the overall process generally remains the same. Okay? Now this N-glycosidic bond will look into these in DNA, RNA, in which you have that sugar, that ribose sugar, and that ribose sugar or deoxyribose sugar is actually making an N-glycosidic bond with a base, a purine or a pyrimidine. We will see those bases. So we hinted at those. So adenine, thymine, guanine, and cytosine, they were all nitrogen-containing compounds. And all these nitrogen-containing compounds are called bases because they can donate electrons. So that's what they're Lewis bases. So that's why they are called bases. And these will have either a 6-membered ring, which is called a pyrimidine. So you will have 2 types of pyrimidines. You can have purines. We will look into that. But essentially what is happening is this sugar is forming an N-glycosidic bond with a base in the DNA. And that is what is forming the main structure or the backbone of the DNA is through this N-glycosidic bond. Okay, we'll look into that. Okay, so this is it for sugar at the moment. Let's move into another class of organic or biologically interesting organic compound called the lipids. Okay, so in lipids you will find again like different kinds of lipids. So some of them will be chained, some of them will be ringed. But typically what these lipids you will find the commonality will be they will be long chain carboxylic acid. That's what lipids are, typically, like very, very simplistically, okay? These are also called fatty acids. You could have saturated fatty acids, or you could have unsaturated fatty acids. Something very similar to this. So this is a long chain, so they have a lot of carbons, and then you have a COOH group. So this is an acid, but this is called a fatty acid because of this long chain. Tell me whether this is saturated acid or unsaturated acid. What brings in unsaturation factor in an acid or in a hydrocarbon, as we saw? What will you see in that molecule that will tell you whether this thing is saturated or unsaturated? Or double bond, triple bond, or rings. Do you see any of these things here? So what will you call this? This is a saturated acid. But the moment you start having rings or double bonds, triple bonds, those kinds of things in here, you will get unsaturated acids. Okay. This similar to this, so I have another structure. So you can have some double bonds in here, stuff like that. You will call that a free fatty acid because this is just that chain. Okay, and this can act as an energy storage molecule. Okay, there could be different functions of the fats, okay, or the fatty acids. Energy storing is one of the most important functions of a fatty acid. Okay, so you can store a lot of energy in fatty acids because you have to break all these bonds and then you can release that energy. So it's that long chain is what is containing that energy for you. Okay, now with this what is this compound? Or what is the functional group of this compound? This particular compound is called glycerol. And we'll see a lot of involvement of glycerol with these fatty acids. Now remember esterification, in which you can have a COOH plus this water molecule can come out and give you an ester. A similar thing can happen. Just imagine this entire group is coming in and interacting with one of the OH, because essentially what will happen? This OH and this OH and this. So imagine this big RCOH group with one of these OHs, another big with another big of the, another fatty acid coming in for this, another fatty acid from this. So essentially what will you end up having is something like At this point of time, I'm not counting the carbon chain, okay? So from that glycerol, you can form these kinds of compounds in which all these fatty acids are coming, individual free fatty acids are coming with your glycerol and forming this huge molecule. We call them triglycerides. This is triglyceride. You probably have heard these terms, the triglycerides, right? Where do you see these triglycerides? Animal fat has a lot of this triglyceride. Plant oils will have triglycerides. Make sense? Okay, now in general what we'll see that life typically has 12 to 22 carbons, and specifically a lot of these will have 14, 16, 18 carbons, like in those lipids, in these fatty acids. A lot of these will have these numbers. Now, first of all, if you don't have life involvement, it becomes really difficult to make these big molecules. Life has to get, so like sometimes these molecules act as an indication of life because A, it is difficult to make these big molecules, big polymers. This is not a polymer, but a big molecule. It's difficult to make, so life helps in making it. And the second is they are very choosy about. So they don't make any. They are making 12, they are making 14, they are making 16. So they are very typical. So those typical compositions are actually helping to, or may help in actually saying something about the presence or absence of life. Okay, let's look into the structure again. What is also interesting to see is how this part is different than this part. This part is hydrophilic, meaning this is water loving. Why will this be water loving? Because of the COOH group. So this is the polar group, polar side of things. Okay, because of that COOH. Water is polar, this is polar, right? So there will be some interaction with that. So that's why we call it hydrophilic, because this will love water because of this dipole thing. So like dissolves like, this is a very famous thing. So polar things will be dissolved in polar solvents, non-polar solutes will dissolve in non-polar solvents, right? But this thing is behaving very strangely, right? This has a hydrophilic head and a hydrophobic tail. It has both these things, and that's why we can also call it an amphiphilic substance. Or has an amphiphilic behavior. We will see how this particular thing helps us in creating the cell membranes and stuff like that. But before that, let's also look into another kind of a lipid, which is called a phospholipid. The phospho is coming from phosphoric acid. So you can think of this. So phosphoric acid is what? OH, OH, and OH. This is phosphoric acid, S3PO4. It has 3 acidic hydrogens, correct? Right? You can see them, right? So all these are acidic H's. Now just imagine that this OH or this part was involved in with something like this. We'll look into what these actually mean, where they are coming. But you can imagine that there is an amino group or something like that can come in and do this thing. And you can have this OH stays the same, but this OH is involved in an esterification kind of a thing with a fat. Now, this was the glycerol base, correct? Now, this glycerol base is attaching itself to a phosphorus group, the phosphate group. That phosphate group in turn is attaching with something else. But you're also getting this lipid kind of thing in here. So this becomes, so this was very similar to the triglyceride that we saw. So this was the glycerol and all these chains of COOH, all these were attached. So this was the triglyceride group. This is the phosphate group. So that's why we call them a phospholipid. So this was that lipid part. Triglyceride is a type of lipid. And that lipid is attached to the phosphate group. This is giving you the phospholipid. We will see how these phospholipids become really important in like cell assembly and things like that. The unique thing becomes this hydrophobic tail and hydrophilic center. What this will happen is, we'll also probably have heard somewhere. So all these different tails will come together with this hydrophilic part. Right? So you can have all these So this is the hydrophilic part, all coming together. This is the hydrophobic part. What can happen is, this can, in our daily lives, when your clothes get stained with fat, it gets really difficult to remove that. Why? Because of this. Because what that fat is doing is bringing in all that dirt together and forming this closed core. This entire structure becomes so stable that it becomes difficult for us to break it. That's why if you get stained by fat, it becomes difficult to clean it. This can happen in many different, but this is one of the life kind of situations. You have to use something to break this. What will you have to use? Soap and hot water. That